I should say that when Aviv and Regina joined me as being organizers of this, I had to prevail upon them to speak as keynotes. And was that a good decision? So we now turn to the third of these remarkable transformations of computation in cancer. This is Regina Barzilay, who's a professor at MIT in MIT's Computer Science and Artificial Intelligence Laboratory. She is a long-term innovator in machine natural language processing and deep learning in chemistry and oncology. She has received many honors as well. I'll just mention one. She was selected for a MacArthur Genius Fellowship in 2017, and she was elected as a fellow in the AAI in 2018. Regina Barzilay. Regina. Everybody?
Well, hey, I was so stressed that it's not going to work, but it worked, so it's a good beginning. At any rate, so very good to see you here. And I'm going to slightly switch gears and talk about technologies that face patients today in the hospitals. And I will talk about clinical oncology. And this is a really interesting area when you're thinking what's going on today. Whenever you are opening a newspaper or Google News, you would see tons and tons of stories about the miracles of AI and how it cures different patients. But in reality, if you're looking at the real penetration of this technology into hospital, it's actually very modest. So according to one survey, only 5% of the US healthcare providers are claiming that they're using AI. So I hope that we are at the point where these changes will be much faster, and there are lots of good reason to believe that machine learning can totally change the way we're thinking about the disease. Let's just think for a second how today we are mapping the patient to a cure. So when the patient comes in, we collect a lot of data about the patient, be it radiology, be it pathology. These images have hundreds of thousands of pixels, and they're really unique to your tissue. However, after that, we have a human who reads this image and summarizes it as a story. And all these unique features of the patients are already lost because we summarize very rich information in just a one page of writing. But this is even not what we use to decide about cure, because we further distill this information into a few categories based on which we determine cure. So if I am to make an analogy about Amazon, you can think that we take the whole US population, divide it to 100 bins, and sell to each one of them the same product. And here you can see it in more clinical terms, that if we look at 4 women who have triple negative cancer, if you look at their pathology, they look very, very different. However, in reality, they are all called triple negative. So as a result, if 3 of these women were fine and 1 of them gets recurrence, there is no way for us to reason about it at this level of abstraction. And then there is another slide that I want to share with you, which is a very troublesome slide. And it comes from the American Society of Clinical Oncology. And we will have some representative from this organization speaking to us later, which says that today, all the clinical decisions in this country in oncology are based on 3% of the population that participate in clinical trial, which means the vast majority of the population for which we know outcomes are not used to adjust the care. And in some ways, it is not very surprising, because today, if you want to use retrospective data, there should be a human who sits there, who summarizes the data, put it into a table which computer can analyze. And just to give you another source of information, all the cancer registries in this country today summarize information about the patients by hand. And this totally doesn't make any sense, because in all other area of computer science, natural language processing is widely used to do this kind of computations. So the way I see machine learning can really change the way we are diagnosing and treating this disease is thinking about it as a probabilistic problem. You are given all the raw information about the patients, be it pathology, radiology, sequencing, and from there you want to really predict the outcomes, population at risk, and the treatment. So the first problem when you're thinking about this view to actually do it at scale is to extract all the information about the outcomes that is currently all in natural language. And we definitely are not thinking about doing it by hand. So we have demonstrated the feasibility of fully automating it at MGH, where we took all the reports about breast cancer patients and we summarize this information fully automatically. So this system, which achieves today the accuracy of the human, can take the report and extract all the biomarkers of the disease. It can extract the lymph invasion status and many other things. And the interesting part is how this system, which is really simple from natural language processing viewpoint, can totally change the care. So for instance, New England Journal of Medicine published a few years ago a paper about precancerous condition called Atypia. The New England article only used 674 pages. You also see reference to JAMA paper, which used even less patients. You can ask why, if the disease is so common. And the reason is that the doctor had to go by hand through 10,000 patients to identify the one which had the right type of atypia and didn't have prior cancer. Today at MGH, with a single query to the system, you can identify 6,000 patients which have this condition. And we can totally see as we move forward our ability to access this data with natural language processing, which really changed the scale of analysis. So another example, and I will talk about it more later, is actually using deep learning for radiology. This is maybe one of the most successful examples of penetration of deep learning, and I'm happy to say that the systems that we developed jointly with MGH has been in use for 18 months, and here you can see Connie Lehman, who will be our latest speaker today during the panel, where the machine read the image and Connie read the assessment and pressed the button. We'll talk about it in more detail later. And the final thing, which is not really interfacing patients, but I'm really excited about it, and I want to share with you like a small preview, is an area of using deep learning for drug design. And when you're thinking about it, the problem of drug design, it's totally machine learning prediction problem. You have a huge space, combinatorial space of molecule, and you need to predict which one of them are promising candidates. And here at MIT, we're doing a lot of work on looking at different aspects of drug discovery, like predicting properties, modeling biological and chemical properties, and finally, maybe the most exciting among them all, using machines to generate new molecules. When you start with the beginning molecule, and you can optimize it to have the desired profile. And Tommy Yakala, one of the speakers later, will tell you more about this achievement, but I'm really happy to share that we started recently our mice experiments, and we really got promising results. So we're working on this topic with the members of our pharmaceutical consortium, and I believe this will be one of the fast-growing areas where AI can really make a big difference, and is making a big difference. Now, I want to spend my time talking about one specific application of machine learning to cancer care. And the reason I selected this one, not only because it works, but because it can really demonstrate to us different views of how communities think about it. And what I will focus on is not the task. Can I actually use it? Oh, it works, fantastic. I'm afraid to touch anything. Oh, I cannot go because I don't have a clicker. Okay, we're low tech here. At any rate, so the task that I'm gonna look at is predicting breast cancer in the future. So there are a few radiologists here that I know about. They should agree with me that if you look at these images, none of them have cancer now. But one of these women actually developed cancer within 3 years. And the question we want to ask computer, can you look at these images now and do the task that radiologists cannot do? Can you tell us which of these women is going to develop cancer? And the question of risk in breast cancer, where there is a large-scale population screening, is really, really important one. It's so important that this year, our President Trump actually introduced a new federal law that mandates providers to tell to women about their risk based on the density of their breast. So this brings us to the next question. And before I go to deep learning, I want to say a few words about how humans are thinking about this prediction task. So the history actually goes way before the deep learning. It comes to a fellow doctor called John Wolfe, who noticed that if you're looking at the images of women who developed cancer, but in their prior images, you can actually detect certain patterns. And at the time, he mentioned that you can analyze it by computer. It was in '67, but now in 2018, we can actually analyze it by computer. But American College of Radiology took it in a different direction. They decided that, of course, there are some patterns, and we want to formalize this pattern. So again, we took all these very different images and summarize them into 4 categories, which are called density. And the assumption is that the first 2 categories, which are considered to be non-dense, are fine categories, but the latter 2 actually are considered to be increasing your chance of getting breast cancer and to get false negative. It affects what we say to the patient, and it affects reimbursement and treatment plans. Okay, so then the question is, it's funny to stand with it. Okay, the question is how we can actually translate it into the risk. And that's where we come with our human understanding. We're thinking, okay, we need to understand how risk is computed. Let's think about different factors. Of course, age matters, of course, family history matters. Let's collect all these different factors and produce you the risk. So models that produce risk, if you do it, you get a UC of 0.6. Just to tell you that for those of you who are not familiar with the area under the curve, if you do totally random guess, you get 0.5. Now you can think, if I'm taking this information and add to it something about the breast, about the density, because it's really something unique, how much do I improve? I just improved by 3 points. I moved from 60 to 63. So there were more people in various clinical disciplines. There are people in this audience who wrote their PhDs and papers about computing clinical risk. But the final point today that we really cannot compute the risk of a woman getting breast cancer above low 60s. This is the state of the art. And one story that you can think maybe the reason it doesn't help is because the way we are computing density is not precise. And indeed, there is something about human eyes. For instance, there is a study that demonstrates that if you show a collection of images to radiologists, some of them detect only 6% of them as dense, and others 84% of them as dense. So one of the first things that we've done with Connie Lehman, we actually built a system at MGH which reads consistently the density. Because you have to report it no matter what you think about it. So we've seen that it actually works pretty well. Humans agree with the system 95%, but it still didn't improve the risk prediction. And the reason is that you're taking human biomarker, imprecise human biomarker, and you are wasting a power of machine to compute this imprecise biomarker. And what we've done, I actually selected, I wanted to see how weak is a biomarker that is federally mandated today. So we took 200-something thousand mammograms, which contained around 1,000 cancers, and we divided the population to the dense breast and non-dense breast. And what you discover here is that, of course, there is some correlation. For instance, if the breast is non-dense, there are 6 out of 1,000 cancers. And if the breast is dense, it's 8 out of 1,000. But there are 42% of women which are in this yellow category. So 42% of females are getting a letter that tells them that they're in high risk. Does it really make sense? Not really. We decided forget about human eyes and about human predictions. So let's just redefine it the way how we do it in machine learning. We look at these images and then we train the machine looking at the image to say whether the woman gets cancer or not, because we have an outcomes. And this is my very favorite visualization of deep learning. You know, you're taking the image, which is a vector, you're doing a lot of non-linear transformation, and it learns to predict. In this case, it predicts cat or dog. In our case, it will predict the cancer. And we used a very rich data at MGH, where we know the outcomes of women because the data is related to the repository. We didn't do any exclusions, but only women for whom we didn't have follow-up of information. And I'm showing you this color-coded thing, and we will come back to it later, which demonstrate what it takes to build the system. So the blue one is if you just take whatever is out there in the package and try it. The yellow one is what I expect students that took at MIT 6036, which is machine learning class, to do, which is just do kind of standard engineering tricks to make it work better. The green one is where we really did a computer science, and we build models which are specific to the task, which is, in this case, we are predicting multiple outcomes, and we want to make sure that the risk curve looks like what we expect it to look. We want to make sure that the model generalizes well across different devices and different signal processing algorithms. You can do a lot of adversarial training to achieve it. And finally, the technical question is how do you combine multiple views, because for every breast you have many views, and typically women have 2 breasts. So there are all the technical questions how you can actually put it all together, and we've done that. Let me just share with you the results. So what you see here in blue is a model that in many hospitals in this country is used to decide who is high risk. It's a one of the top performing models, if not the top performing models. You can see that predicting 1 year risk, this model does 0.66. The image-based model achieves 0.88. And now people are asking, are you comfortable with 0.88? You know, it's a question if I am or not. But the point is that we are now clinically deciding the fate of those women based on 0.66. Now, I've seen some papers, actually, Connie sent me, which I find very entertaining, which talks about the radiologist insight and intuition. And this particular paper says, you know, radiologists actually can predict who is going to get cancer looking at the image. The only problem with it, that IUC reported in this paper is 0.53, which is a tiny bit above random. I mean, I guess it does say we have intuition. You can see that this model does, when we are looking on 2 year risk, as we would expect, it goes down, but it's still 0.81. And if we combine this AUC with a C index over 5 years, we can still see the deep learning model really performs very well on this task. And the point that I want to make here, that what we've discovered, the density and high risk are not the same thing. You can have high density breast with low risk and vice versa. People in radiology knew it anecdotally, but we really demonstrated this is the case. So I'm not sure what was the value of this new federally mandated biomarker now. At any rate, and coming back to the presentation that Phil was given about pancreatic cancer. Of course, you don't have to only to ask this question about breast. We are currently expanding it to looking pre-screening images in the pancreatic cancer and lung cancer. So there are lots, we can already imagine the change that is coming, that for many of these diseases that we historically diagnose them later, we will have the capacity to diagnose them much earlier before they actually develop. Okay, now I have 6 minutes and 54 seconds, and I want, as a machine learning person, to talk about, my observations about myth in plural, of using AI in this area. I want to start with the first myth, which so very eloquently was said by the Chief Data Science Officer from Partners. So this myth relates to democratization of AI, where he tells us the truth is you don't have to be a computer scientist or data scientist to participate in the creation of AI. We are just starting to see increasing availability of tools to enable on-premises development of AI models by clinicians. So it's almost like saying, Regina, you can cut at orange, you can also go and cut breast because you know how to cut. And I want to tell you, the reason I color-coded to you the pictures before about our model, I say, again, this is a model that, if you know a bit of computer science, you can program, you can just take the package and run. This is if you kind of passed a reasonable machine learning class and you have some experience. This is really a custom build with an understanding what the model should be capturing. And you can really see here a big difference. If you just take some tool and apply it, you get something, again, 0.57, a bit better than random, but not much better than random. And you can really see that there is a lot of value on developing the model when you understand what you are modeling and you have the right computational tools. And I would not bore you with the similar statistics for chemistry and drug design. We see exactly the same. You take a standard model, you just apply it. And I think this is not the problem of medical community. It's more of a problem of my community, of machine learning community, is that we're still not at the point that you can just apply it and use it as electricity. But given the current state of art, I strongly recommend you look at these numbers and then decide, do you just want to take something and apply it, or maybe there is a place to really do their computer science. The second myth, this one was again eloquently stated by a doctor who wrote the story about it in New York Times, and it says AI could worsen health disparities. And just tell you about the health disparities, I would show another numbers related to the data that I presented about risk. So now I want you to look at the stratification of the performance across different cohorts of population: white women, African American, and Asian women. As you can see that for non-white women, the model, the hierarchic model, which is currently used in clinical practice, performs very close to random. And this is not some exceptional story. We can see it across the board in many, many other applications when clinical trials were developed primarily for white men in the past. And this hierarchic model is today clinically used. So what I strongly believe is that we have now an opportunity to change it. And we can see that the deep learning model actually sustains performance across these different cohorts. And there are lots of things we can do within AI to make sure that we don't have bias. We can do proper data curation when our data is representative of the population when we are trying to apply it. And it can be maybe Asian population in one place and Indian population in another place. We can just easily retrain these models. Then there is a second point, it's anti-bias training. And here I'm not talking about HR kind of training. I'm talking about developing machine learning models that can be trained in such a way that they sustain performance across different subpopulations, even smaller subpopulations. This is a big area of core machine learning algorithm that can absolutely be used in this area. And the final point, that of course we need to do human-machine validation to make sure that we are not introducing extra bias. The third myth has to do with black box architectures. And there are quite a number of people, which is sort of entertaining, which are not computer scientists, who identify this problem and try to solve it. So let me first tell you, there are lots and lots of methods out there in machine learning that enable you to interpret. And we will get back to it, what does it mean to interpret. And then the second myth that is hiding there, it's like 3.A, interpretability is key to clinical adoption. So I should tell you that correlation-driven care, I think, is really a big part of cancer research. And if you don't believe me, just look this beautiful account, The Emperor of All Melodies, a biography of cancer, and see how many things we are doing just based on correlation. And maybe density that is currently used is prime example of that, but there are many other striking examples. So not understanding the mechanism in clinical care is actually many times it's a big part of the story. Again, I'm not suggesting that's how we have to move forward. So at MIT, we've done a lot of work on building interpretable models, and there is a question again how we can interpret. It can demonstrate to you which part of the input it looks at when it makes its decision. It can find you very similar examples based on which it makes its conclusion and so on. But to me, the question, and it relates to the presentation that Aviv gave, what exactly does it mean mechanism? And I think there are different ways to think about mechanism. For many tasks, machines are already stronger predictors than humans, and predicting risk is one of them. And the question is, can we really understand the underlying pattern? And maybe if we will force machine to explain to us its pattern in a way we can understand, we can actually oversimplify and bias this pattern. So I think there's really a question when we are thinking about explanation, what is a reasonable way to formulate it? It still glean some insights of what's going on. And the final 34 seconds I want to spend talking about the J-Clinic. J-Clinic, it's a new initiative at MIT on machine learning and health. Here you can see people who are involved in this initiative, and Phil Sharp is the chair of our advisory board. And the way we are thinking about it is really to bring together computer scientists and biological engineers, biology, and everybody else who cares about health on campus, and focus on development of algorithms such as reducing need for large amounts of annotated data, privacy-preserving learning when you are encoding the data and learning on encoded data. How can you do robust transfer across populations so that you don't need to train for every group separately? And finally, to think more about what does it mean to make this model transparent and interpretable. And a big part of it, of the J-Clinic, is taking this algorithm that we are developing and not just to try it on one institution, but try it broadly across many places, and we are now actively recruiting hospitals that are going to be part of our network, that based on what we are learning, what happens in practice with the algorithm, we can update the algorithm accordingly. So thank you very much.